Okay, so this work that we have performed represents a small step in our long-term ambition to create plants that are capable of fixing atmospheric nitrogen. And the background to this project is that biological nitrogen fixation that takes place in the nature is happening by a set of prokaryotes. So we have bacteria and archaea. And what we mean with biological nitrogen fixation is the conversion of atmospheric nitrogen into nitrogen that is biologically available, such as ammonia and nitrates. So, and these levels of available nitrogen in the soil is actually one of the main limitations for the crop production. So how we have solved this in developing countries is by the use of synthetic fertilizers. And of course this has unfortunately very negative effects on the environment because of two main reasons. First of all, the synthetic fertilizers takes a lot of energy to produce them, and another main disadvantage is that a lot of the nitrogen in the fertilizers is actually not taken up by the plants. It is leaking out into the soil and into the water. On the other hand, for the poor countries, the developing countries, underdeveloped countries, for them there are of course economical reasons why they cannot use the fertilizers. So what we are trying to do in the group of Luis Rubio is to transfer this prokaryotic nitrogen fixating machinery into plants, to make the plants capable of taking up the atmospheric nitrogen. This will then have, on a long-term goal, if we succeed, will have very positive effects on the nature, but especially important that this will have a positive effect on the poor countries that today cannot use fertilizers. And this is one of the reasons why we are supported by the Bill and Melinda Gates Foundation. So to explain the project more on a molecular level is that prokaryotic nitrogen fixation takes place using two protein complexes. So we have one protein complex that is the denitrogenase reductase, which donates electrons to another protein complex, which is the nitrogenase. So the nitrogenase uses these electrons to break the triple bond of atmospheric nitrogen and converts it into ammonia. However, to make it even more complicated is that for these two protein complexes to work and to be matured, they need the help of other accessory proteins, and this is a rather large set of accessory proteins, around 10 other proteins that are needed. So the job of these accessory proteins is to help mature the nitrogenase proteins, and importantly to synthesize metal clusters that are absolutely required for the activity of these enzyme complexes. So this work, what we have done in this paper, is a collaboration with Professor Christopher Voigt at MIT, which is a world-leading synthetic biologist. So basically, what we have done is to create a library of yeast strains, so about 100 yeast strains that expresses 9 nitrogenase genes. The reason why, so these 9 proteins would then represent the kind of a minimal theoretical amount of proteins in order to obtain a functional nitrogenase. So what we found in this work is that we could actually express all 9 proteins in a eukaryotic cell, which is very important. We could also target them to the mitochondria, and we could prove that we have a nitrogenase tetramer formation within the mitochondria, which is an absolute requirement in order to obtain a functional nitrogenase.